All right, thank you for the introduction. Next time I submit one of those, I'm going to have to shorten that bio a little bit. So thank you everyone for being here. As you heard, I'm Paul Schaffer. I'm the Director and Division Head of Life Sciences at TRIUMF, Canada's National Particle Accelerator Facility. And today I have been tasked with walking you through some of the preclinical development aspects of isotope research and development, but also chelate research and development, or more specifically around radiopharmaceutical development as a whole. So hopefully walking away from this, you'll see a build on from what Dr. Zakir and Dr. Silivanova were speaking about. Before I begin, I do have to disclose that even though I am a full-time employee of TRIUMF, TRIUMF is part owner and I am compensated by a spin-off company called Artemis. I am a listed inventor on some of the technologies licensed to Artemis. And this presentation will mention some technologies that Artemis is marketing, but to avoid bias, I will do my best to present advantages and disadvantages of alternative technologies to that particular piece. So as for today, the topics of the discussion will be to give you a brief overview of what TRIUMF is, its capabilities. There will be a twofold discussion around isotope production research from more of a cyclotron perspective. And then I'll also close out with some discussion on radiopharmaceutical design and development and provide some perspectives in my summary and conclusions. For those of you who haven't been to TRIUMF, this is the laboratory here. You can see it on the southern tip. It's the complex of buildings on the very southern tip of the building mass that you see in the center of the picture. That's the University of British Columbia, Point Grey campus. And it is obviously in a beautiful setting. It's been there for over 55 years and has long been home for particle and nuclear physics research in Canada. I think it goes appropriate for me to say and mention at this time as well that UBC and TRIUMF sit on the traditional territory of the Musqueam people, where they have used that land for learning since time immemorial. And I wanted to acknowledge that here to this group today as well. Inside of the accelerator facility complex, you'll see in the main accelerator hall a series of equipment and shielding arrangements that look like the photograph here. This is a 1968 origin technology. The world's largest cyclotron sits underneath the yellow blocks with the people standing around it in the center of the photograph. The beam lines extend for tens of meters out of that cyclotron. The main beam line, which is actually going to be the subject of some discussion today, and the production of some of the isotopes you'll be seeing, extends toward the bottom left. And from there, it's just worth mentioning that TRIUMF is home to over 600 staff and students on any given day, with over 1,000 visitors from around the world coming to do primarily fundamental particle physics and nuclear physics research. The good thing about life sciences program in a facility like this is we get to use all the stuff that nobody else wants. It's the long-lived isotopes that have medical application, and that's what our group subsists off of. If you peel away the roof and turn the facility into a schematic, this is what you'll see. The main cyclotron you saw underneath the yellow blocks is kind of in the center bottom of the schematic. The main beamline you saw projecting to the bottom left, again also under blocks, is kind of shooting off to the right in this particular photo. And that cyclotron drives a number of other experiments to the north, which are affectionately called Ariel, or the Advanced Rare Isotope Laboratory, or ISAC 1 and 2, which are the isotope separator and accelerator facilities. And you'll hear a little bit about what those are doing in today's presentation. So overall, 6 cyclotrons, soon to be 7 cyclotrons, and 2 linear accelerators are on site. We have over 1 kilometer of beamlines, again, primarily focused on basic research. We have collaborators from over 40 countries, and that number over 1,000 people before the pandemic hit in 2020. The range and energy of these cyclotrons, you might wonder why we need so many. They actually go from about 13 MeV up to over 500, and that gives us access to a range of different isotopes as well. Coupled to that is different beam intensities from each one of those machines. The 13 MeV machine has long been used for our positron emission tomography isotope production program. We also have commercially used cyclotrons, 2 TR30s and a 42 CP42 operated at 30 MeV, owned by BWXT, operated by TRIUMF. And those are along the right-hand side. You can kind of see them silhouetted in the building complexes on the very eastern side of the campus, with the TR13 being the fourth one on the bottom there. The main cyclotron, as I mentioned, is in the center at 500. And from there, the focus of those cyclotrons is isotope production, radiochemistry, but we also do some exploratory basic research in proton therapy and bio-beta NMR, which is an analytical technique that utilizes beta decay to understand the magnetic properties of matter. So in Dr. Zekier's presentation, you saw a chart of nuclides. That was that pork tenderloin shaped list of neutron and proton based elements. And if you collapse that back down, you get the periodic table again. And I've color coded it a little bit to show you all the different types of emissions that you can get from some of the isotopes. So that depth in the neutron axis is actually now represented in colors. And you can see there are quite a few positron emission or emitting isotopes, a number of single photon emitters. But then there's also a growing recognition for the number of particle emitters in the form of beta, alpha, and Auger electron emitters. So across this particular periodic table, I think maybe it's worth mentioning that over the years, most of the focus has been in the upper right corner, and the main group positron emitters in the form of carbon and fluorine with some nitrogen. You saw some talks earlier today with nitrogen-13 involved. And those dominated the field of imaging for a long time, with technetium, the green box in the center, dominating the SPECT world for a number of decades as well. We're starting to see an expansion of that out and away from technetium and the main group into some of these other metals. And today I'm going to be talking to you about what's involved with doing that. So radiopharmaceuticals will solidly into what we call a generation 4 paradigm now, where you have molecularly targeted entities that are tethered to an isotope of some kind. You can see a graphic of that on the left-hand side, where you have that biomolecule that targets whatever it is you're injecting to a specific tissue. There is a linker, and then there is some form of chelator that can bind to the isotope of interest. That's significant because you notice that most of the isotopes that were beyond the main group were all metals, and metals rely on chelates in order to be controlled in a chemical form. And as the community transitions from an imaging focus to a therapy and theranostic focus, that's the Gen 4 part of what I just commented on. These chelator and targeting vector combinations are becoming more and more important. And so we do a lot of research at TRIUMF and with our partners on exploring the chemistry involved with those chelators and tethering them for achieving the results we want to see in the clinic, which is represented on the right, where you see a series of lutetium images of a patient undergoing treatment for prostate cancer with an image courtesy of from Dr. Francois Bénard at BC Cancer. So what is helping metals become more mainstream is the advancement in a lot of technologies around producing them. Now, this is in particular recognition of more mainstream cyclotron centers located in hospitals around the world. I don't want to diminish the contribution that advanced centers that have operated more powerful cyclotrons for decades in some cases, that what they've contributed. Those centers were specialized and they were very good at what they did. But the technologies around making metals on an everyday basis in an everyday cyclotron center are becoming more and more easy to obtain. And so back when I first started at TRIUMF almost 15 years ago, I was responsible for leading initiative to both develop high-power targetry, shown on the left, coding techniques and radiation parameters for different target types. You can see an elongated target in the second panel for producing technetium-99M by cyclotron. In the center panel, a circular target used to create gallium-68. We partner with robotic automated synthesis unit manufacturers to then develop standardized chemistry to isolate and then formulate the different products. So really the result of all of this technology coming together is something that is giving a standardized and automated, repeatable, robust way of making radiometals on a day-to-day basis. And the reason why that's important is shown here. So I show an example of just one isotope. There are many examples. Every isotope you make can have its own set of examples. But what you're looking at is a graph of cross-section or relative probability of proton capture against the energy of the protons that are striking it on the y-axis, And what you can see from the 3 vertical bars is that different cyclotrons are made differently. And so you have the potential of irradiating these targets under slightly different conditions. And based on the different products that you can see, you can strike a target that is made of molybdenum 100 and make 3 or 4 different products, as you can see here. So if you have a slightly different cyclotron energy between one center and the next, you have a potentially different quality product that emerges. So when you're trying to roll this out for clinical use, and the idea is you want standardized quality, you need to choose the parameters that you want to irradiate. So from there, you are standardizing the irradiation conditions in order to allow for every center, regardless of where they are, to achieve the same things like specific activity, which is the amount of radioactive to non-radioactive material, the purity, and then the quality control, rapid testing requirements, and knowing that they will perform as designed. And then ultimately, the kits that a lot of companies make to formulate your radiopharmaceuticals will perform equally as well as their shelf life. And then, of course, ethical considerations around patient dose and clinician enablement through image quality. The list of publications at the bottom really stories a 10-year effort that describes the production of technetium by cyclotron and all of the travails involved with that. The hardware can be used in relevant adjacencies, and so technetium is shown on the left, but this was quickly evolved to produce gallium, copper 64, zirconium 89, and we've recently been able to demonstrate significant quantities of production quantities of each one of these isotopes, again under conditions that are growing to be more standard across different centers. So with terabecquerels of technetium that can be produced, you can also make hundreds of gigabecquerels of gallium and onward from there. The longer the half-life, the less you make. But nonetheless, this is an emerging capability across a number of cyclotron centers. And just to compare what advantages this have using gallium as one example, on the right-hand side, you can see cyclotron production can produce up to 10,000 millicuries of gallium in a 2-hour irradiation. It is comparable to generators, which are coming on 100 millicuries per elution on any given day. So you can see the substantial production quantity differences there, but also those relevant adjacencies I mentioned and the ability to make other metals, whereas a generator, you get one isotope and that isotope that it's designed for. The risks are different. Widespread outages can happen on generators because the source isotope is typically made in a large producer like a reactor, not always. And then on the cyclotron side, you do have a decentralized paradigm where you have a greater risk for variability from one center to the next and a greater skills burden on any given site to do things properly. So that was the focus on the diagnostic isotopes that have permeated the community over the last several decades. Where are we going? As you heard in the previous talks, we're heading toward a theranostic therapeutic paradigm. I'm not going to go through this in great detail here. You've heard about it in the earlier talks. But moving toward prostate and neuroendocrine treatment examples, you see 2 with actinium in the left and in the center, and 1 using lead on the right. And those Gen 4 radiopharmaceuticals are shown chemically on the bottom below each one. So those are the compounds that are responsible for the therapeutic outcomes and delivering the isotope to the tissue of interest. And I'm just going to go forward 1 and come back 1. So how we do the exploration of isotope development at TRIUMF, the 500 MeV cyclotron was used to irradiate thorium-232. This began in 2016 and merged into a campaign this year. We don't have all the results just yet, so those are forthcoming. But we're actually able to produce actinium in 2 ways, and I'll explain that in a second. One that has some 227 in it, but also some that has no actinium-227 contamination. And what happens when you irradiate thorium-232 with 500 MeV protons is you actually make 300 different radioisotopes at the same time. We actually developed the chemistry to extract just actinium from that. And so those colored boxes in the periodic table show actinium on the bottom left is orange, meaning it's the only color that was in that fraction. Then there was also the yellow fraction, which has a few of the lanthanides above it. You can see green and purples, and those all existed in co-elutive fractions. So this is just to give you an idea of how complex the chemistry can be around producing an isotope like actinium from a heavy element like thorium-232. And as I mentioned, we can produce 2 flavors of actinium. On the left, we call it directly produced actinium, where you have the immediate spallation mixture Take all the short-lived actiniums away, you're left with 225 and 227. But we have a high enough energy at TRIUMF to produce also radium-225. It's in a series of radiums, but fortuitously, it actually is radium-225 is the only one that decays to a meaningful actinium product. And so we can actually generate actinium with no measurable amounts of actinium-227. And I'm proud to say that this approach has now been adopted by BWXT and is being used in clinical trials as we speak. Moving forward, big machines like the one at TRIUMF are not going to be able to sustain a global supply of that isotope. So the future is very much in the lower energy approach. You heard a little bit of that from Dr. Solivanova's talk, and that's going to be the low energy irradiation of radium-226. And more information, I'm sure, is going to be coming out on that as more developments are made in the months and years ahead. The benefit of splitting thorium, as I mentioned, is you do make a mess. You make 300 different isotopes, but you can start to pick out some of those for additional research. So you heard about the center column, which is the radium-225 and the successive daughters of actinium downward. But we're also looking at things like thorium-228 on the left and protactinium-230 on the right. These can lead to their own daughter products that also have potential medical application. These can be paired then with lower energy processes that make the imaging isotopes you've heard before. In some cases, such as the production of lead-212 from the decay of thorium-228, it can be paired with lead-203 as a theranostic pair, both for imaging and for therapy. So these are some of the applications we're looking at. And the reason for that is, again, just to remind you, the use for lead-212 for treating neuroendocrine cancers. And excuse me, the example shown again as a reminder on this slide. So to finish off my presentation, I thought I would just quickly go through what's involved with making these radiopharmaceuticals. You see a lot of flip-flopping between, well, we'll just image with gallium and then we'll just do therapy with actinium. It's not quite that easy, at least from the chemist's perspective. There are a couple score of chelates, which are those chemical entities you see on the right that have been developed in the Vancouver area, obviously hundreds across the community. But these are different configurations of those heteroatoms, the nitrogen atoms and the oxygen atoms you see configured around those carbon vertices. And we have basically made degrees and careers out of people stringing together these heteroatoms in different configurations to help understand how they interact with different metals. Things that matter are the ionic radii and different chemical considerations for each one of those metals. And I give a visual representation of how different an element like gallium can be from something like actinium. So it's not quite as simple as swapping one out for the other when you're looking at chelate. And I want to draw your attention to the bottom right. There's one we affectionately call crown. We've looked at that one in more detail. The graph on the left shows a concentration of binding actinium when you're using crown versus the commercially available DOTA. And what the line going further out to the right means is that it has a lower concentration capability of binding actinium, which has implications for radiopharmaceutical synthesis. We're looking at that chelate in terms of how the pH affects binding, with some around the mid-range of that pH 4 to 10 range is more optimal. And then, of course, the amount of time you need to incorporate the metal. So this is important to know, because as you go to use this chelate for radiopharmaceutical development, you need to understand what space you need to operate in. And the example shown here is a crown targeting agent held up next to a DOTA targeting agent, the same one. And great, somebody has a laser pointer going for me here. Great, thank you. Okay, so that's not me. So anyway, so that's great. So the one on the right shows that there, if you look at the tumor with the blue arrow, there's about 6% ID per gram, versus on the left is 12% ID per gram. So it's the same targeting agent, different chelate, you get a significantly different uptake. And that plays into the fact that crown behaves differently when binding to actinium. From there, you can also see the formulation of actinium is something that you need to monitor. You heard Dr. Solivanova mention monitoring for quality. And over time, you can see the alpha emissions slowly degrade the integrity of your radiopharmaceutical. As the series of chromatograms go down, you can see that center radiopharmaceutical peak start to degrade. And then from there, we also, of course, like to do preclinical animal studies and trying to determine whether or not the radiopharmaceuticals under development are holding the effect that we want. The graph on the left shows survivability of animals that have received between 30 and 120 kilobecquerels of injection. And tumor sizes versus body weight are shown on the graphs on the right. So animals receiving 55 kilobecquerels injection survive the longest. Okay, and just to wrap up, equipment like what you find at TRIUMF, excuse me, is novel, it's unique, but it can be used to look at isotopes in advance of them becoming mainstream. So there's a list here in which our 500 MeV cyclotron is used to irradiate different targets. We produce different products. We actually spray paint them onto little targets like you see on the bottom right. We can then do some chemistry and study them. And so examples include exploring the use of a terbium-155 version of that targeting agent you saw on the previous slide with actinium. We can image this with our trimodal, excuse me, PET spec TT machine located at UBC across the street. And from there, assess dosimetry and feasibility of using that isotope for therapeutic application. Or in the case of actinium-226, not 225, we can actually explore the therapeutic benefits of that isotope in the absence of its sister isotope. Showing very quickly on the bottom left graph, you can see the comparison of actinium-225 to 226 dosimetry, where 226 is the blue bars and lower, so therefore requiring a higher injection in order to afford the same therapeutic dose. And the advantage of actinium-226 is that it does emit its own photons, and you can image it directly, as shown on the panel on the top right. Okay, so I'll close out just by mentioning some exciting research efforts that are coming in the years ahead. A team led by Professor Ramajita and Francois Bernard are leading a national consortium in a New Frontiers for Research Fund research effort across 5 different aims to get 3 different radiopharmaceuticals into the clinic, therapeutic radiopharmaceuticals. So we're excited about what this will lead to for the Canadian research community and potentially the clinical community in the not too distant future. With that, I think my voice is just going to have to say, have a look at this slide. There's lots of isotopes that are coming down the pipeline. There's lots of potential coming by way of therapy into the community. There's a lot of risk in supply. We have to stabilize that. We do that by understanding the production parameters, excuse me, and the requirements for radiopharmaceutical production, like what you've seen today. And from there, I want to encourage continued investment in the Canadian infrastructure for making these isotopes, and we look forward to future translation. And from there, I have a large team of people to acknowledge. The names highlighted in blue contributed directly to this talk today. And with that, I want to thank our funders, our partners, and all of you for your attention today. Thank you.